called the machinery, so these are generally part of a complex, sort of. And the HSP proteins, have we talked about HSP proteins at all? Would you tell me if we did? So HSP stands for heat shock protein, remember? And so during heat shock, what happens, right? What happens during heat shock? When you heat shock cells, and you heat shock proteins, what happens to them? They do what?
Denature.
They denature? Sort of, but what happens when they denature? What happens when proteins denature?
They release their bonds.
Hmm?
They release their bonds.
Release their bonds, but then what happens? What happens when you heat shock an egg?
You eat it.
You eat it. But before you eat it,
It changes, like, in an egg, like it's liquid.
you hard boil it, right? And why does it hard boil? What's happening?
Protein changes.
You're heating it, you're denaturing the proteins, and then what's happening?
It loses function, it changes.
The proteins are aggregating, right? So when you heat shock a protein, and you denature it, you expose residues that interact with other proteins, and they aggregate, they come together, they form a hard-boiled egg, right? So heat shocking a protein essentially messes up with the structure so that they'll aggregate. And what the heat shock proteins themselves do, if you take a cell and you heat shock it, and certainly not drop it into 212-degree water, but if you take a cell, you know, a eukaryotic cell that goes at 37 degrees and you heat it up to 42 degrees, you induce the expression of these heat shock proteins. And what the heat shock proteins do is they try to keep the proteins from aggregating, and then supposedly if the cells survive the shock, which is not too long, it helps to refold those proteins. And that's where this HSP was found. There's actually one that's called HSC, which means constitutive. It's HS cellular or HSC, heat shock constitutive protein is made in all cells. It's involved in folding. And it's one of the very first proteins that the ribosome is making the protein, nascent and coming out. There's this HSP protein. And what the HSP protein does is it binds to hydrophobic regions of the protein. So this protein hasn't folded yet, right? It has exposed hydrophobic regions. And if they're exposed, what's going to happen to them? What are they going to do? The hydrophobic regions do of proteins. I'm going to tell you later on love it. If you guys weren't listening. They interact with each other, right? Those hydrophobics, why are they called hydrophobic? Water hating. They can't exist in an aqueous environment. How do they exclude the aqueous environment? They come together with each other. So if you have proteins with hydrophobic regions exposed, they can't exist in aqueous alone, but if they clump together, hydrophobic to hydrophobic, belly to belly, right? Then their hydrophobic regions aren't exposed to aqueous, they're aggregating, they're clumping, they're hard boiling that denatured egg. So what happens is HSP70 binds to those hydrophobic regions, and in doing so, it covers them up so the protein can fold. And you can see here what's happening. Here's a protein that has been made. It's folding, and there's HSP70. HSP70 is an ATP-dependent protein. When it binds to ATP, this guy has ATP bound to it, and as it binds to this protein, it hydrolyzes that ATP to ADP, and that sort of stabilizes the binding there. There's another protein that's called HSP40. Right? HSP40, another heat shock protein, helps the HSP70 bind. So HSP40 sort of loads that HSP70 with ATP on. And then the ATP hydrolyzes it, causing it to clamp down tightly on the protein. So when this guy becomes ADP on it, he's tight on that protein. He's protecting that hydrophobic region so they don't aggregate. Right? But you don't want to protect it forever, ever. You want it to fold. You want these proteins to fold. So what happens is the ATP then exchanges with the ADP, and when ADP binds to this HSP70, conformational change, no longer has affinity for these hydrophobic regions, and it pops off. It comes off. Not necessarily all at once, but they'll come off, and the protein breathes. Right? They pop off here, and two hydrophobic regions of the protein pop back together. Right? Rather than being aqueous, it allows them to interact. Why didn't they do it before? Well, maybe this hydrophobic region that interacts with this one wasn't made yet. So if they bind, and as the protein's being synthesized, what can fold? Synthesis. So it can fold properly, right? Makes sense? But even then, come off and two hydrophobic regions come together, it may be the wrong ones. And there's a hydrophobic regions all over the protein. These guys come off and two of them go like this. So what happens? HSP70 binds again, right? And it causes them to release. And then, so it's a dynamic process with this HSP70 coming on and off, and the protein breathing, right? And all the time it's trying to maximize the energy, or minimize the energy required to hold that protein into shape. And when it finally does, then the process is over. So this chaperone, right? The chaperone, HSP70, is chaperoning the folding of the protein by letting these hydrophobic regions breathe and fold properly. And in heat shock, that's the other thing, right? Where you have heat shock protein where the hydrophobic regions are together in the protein, HSP70 will interact to let them to pull them apart so they refold. So the misfolded proteins refold again. So this is one of the very first proteins that's involved in helping folding. And a vast number of the proteins do. I don't remember the number, but I think that it's like 95% of the proteins in a cell can't fold without chaperones. They don't do it. It's very high. Okay. This is another type of a chaperone. And this diagram is actually one that's found. This is HSP60 like. HSP60 is a protein, is what's called, it's found in eukaryotic cells in mitochondria. In prokaryotic cells, it's called GRO-E, and these subunits are the GRO-E, and that's the GRO-ES cap, and this is GRO-E, GRO-L down here. But anyway, they're called. GRO-EL in prokaryotic cells. It's called HSP in mitochondria or eukaryotic cells. What happened? My battery actually died. How much time? 10 minutes? 5 minutes? This is your chance. Anyway, so here's this folding process here. This is actually a structure that's found in both eukaryotic and prokaryotic cells. It's kind of interesting. It's not an organelle. It's not a membrane around it, right? It's quite big, but what it does is it helps proteins to fold, exactly like we've been talking about. Due to hydrophobic folding. Only in this instance is when this chamber is empty, these ends of the caps here, the barrel, this is an empty barrel, is able to bind to hydrophobic regions. So as the protein moves in there, right, you can see this is bound there. And once it's bound, then these things here are also ATP dependent, kind of like the HSP70. And the HSP, when they bind ATP, it causes them to kind of stretch out and pull out. You can see here. So they're stretched out, they get covered with this GRO-ES cap, but as they stretch out, they kind of pull the protein. So that these regions of the protein that have some sort of interaction, maybe hydrophobic, are pulled away from each other. Once they're pulled away, then they're inside this barrel with a cover on it. And all the barrel does is it prevents this protein, this partially or incorrectly folded protein, when you pull it apart, it prevents it from interacting with other proteins that are pulled apart, right? So it sort of isolates it there, pulls it apart, hydrophobic regions can interact, this guy can try to refold. If it's incorrect, then this cycles, right? It goes through a cycle again, binds to those hydrophobic things there, pulls it apart again, and it goes through this breathing process to allow it to fold once again in the isolation of this cap. Notice it's weird that these things are always sort of a dimeric sort of structure, but it's always one side or the other that works. Once this is loaded, the other one's kind of waiting ready, but it doesn't load also. So this is the second type of chaperone that actually does it in sort of an isolated environment. This is a picture of one from a eukaryotic cell. You can see there's a cap, and there are these two regions here, 1, 2, 3, 4, 5, 6. So these are hexamer donuts that are formed there. You can see that the cap kind of locks things in, and look at it, it's an HSP60 protein, this whole unit. Remember, in eukaryotic cells, it's called HSP60. And the protein that's inside these rings here, it actually gets into that chamber, into that barrel in there. And the proteins, it's in an ATP-dependent fashion, are allowed to breathe inside, and when they're folded, they're spit out. So that's the HSP60 guy that's folded. Okay, so since no one's gonna tell me what time it is, we'll stop there. On Monday, we'll talk about secretion.